Many pregnancies fail early in human embryo development around day 7 or 8 post-fertilization. This protocol offers opportunities to investigators to grow human conceptors in vitro during this mysterious implantation window. This allows us to understand the mechanisms that underpin the success of human implantation and early placenta formation. Using an extended culture system to grow peri-implantation stage human embryos in vitro is probably the only way to obtain authentic materials to study implantation and early trophoblast differentiation. This straightforward technique is a powerful tool that allows us to answer many fundamental questions about human early development. Research conducted with the use of this protocol will largely contribute to the understanding of early pregnancy loss, recurrent implantation failure, and placenta pathologies. Demonstrating the procedure will be Deirdre Logsdon, the PhD student from my laboratory.
One day prior to embryo warming, prepare media and recovery plates in a sterile laminar flow hood. Fill 2 Centerwell organ culture wash dishes with 500 microliters of BM with 10% SPS. Then cover the BM with 500 microliters of embryo culture grade oil. In a 60 mm tissue culture dish, layer 8 milliliters of embryo culture oil and anchor 20 microliter drops of BM with 10% SPS to the bottom. Equilibrate the wash dishes and recovery plate in an incubator at 37.